Hi guys, so we are going to take a look at Gregor Mendel and the introduction to genetics. Hopefully by the end of today, you'll know who Gregor Mendel is, and you'll be able to define these key terms: trait, heredity, genetics, purebred or homozygous, they mean the same thing, hybrid or heterozygous, again, they are interchangeable. Alleles come in two varieties, dominant and recessive. And we have previously talked about DNA and chromosomes. Again, our goal for today is to figure out what controls inheritance in traits of organisms, what forms their genetic code, and explaining the relationship between chromosomes and genes. All right, so the first box that you are filling out, Gregor Mendel, who is he? He was an Australian monk and gardener, and he studied pea plants for over 10 years. Why pea plants? Well, we'll get to that in a minute. What he was looking at were their different characteristics. And he wondered why they had these different traits. That is the key to a good scientist. They're always asking why. And each good why leads to another why, which is kind of interesting. Again, these characteristics or traits were the things that were able to distinguish in his plants. So some of them were tall, some were short, some had purple flowers, some had white flowers. These are all characteristics or traits. He later became known as the father of genetics because, as far as we know, he was the first guy to study genetics in an academic kind of a way. So in class, we took a look at these cats and we asked, what do we notice? And what we noticed was that even though the mama cat is pretty much all white and with very little black marking, we see that pattern not at all in any of her offspring, in any of those 5 kittens in the box with her. One is pretty much all black. All the other ones, even if they do have some white, they have a lot more black on fur than she does. So why do we have these changes in characteristics from one generation to the next? And the simple fact is because cats, like all mammals and like most multicellular organisms, reproduce sexually. Remember, with reproducing sexually, we are always going to have genetic variation, which means that we are going to have unique individual traits from one generation to the next. All right, so Mendel's experiments, what did he actually do? He had the P generation, which is the original parent generation. He crossed a homozygous or a purebred tall pea plant with a homozygous purebred short pea plant. So that is the P generation or the parent generation. That gives rise to the F1 or the first filial generation. When he did that, the only thing that he got were tall plants. So like the shortness trait seemed to somehow disappear. Then he took those tall pea plants and allowed them to self-pollinate. And when he did that, he got mostly tall plants, but he also got about 25% or 1 in 4 short plants. So his big question was, why? Why is this happening? Why does it disappear for one generation but reappear for the next? All right, so why pea plants? This is your second box. Mendel chose to do pea plants because, number one, they grow very, very quickly and produce a large number of offspring in one generation. This makes studying them a lot easier. And in general, the larger your numbers are when you're doing probability and statistics, the more accurate your findings are. Number two, they have many traits which we call bimodal, meaning that they only show up in 2 ways. For example, it was either tall or short. There was no medium-sized plants. There was either a purple flower or a white flower. They didn't get kind of like a pinky, purpley, nothing in between. It was all bimodal. It was one or the other. So pretty simple in terms of genetics. All right, so here are some basic lingo. If you're asked to define these words in the middle of your page. So a trait are different flavors, if you will, of a characteristic. So let's say, for example, A characteristic might be having hair, but different forms of that hair might be brown hair, blonde hair, red hair, black hair. Those are different traits, okay? Heredity is the idea that we can pass these traits from one generation to the next, from parent to offspring, okay? And finally, genetics is, of course, just the study of heredity. So if you are doing any kind of study about the biology of how one generation gets traits from the previous generation, then you are doing a study in genetics. And these are some of the traits in the pea plants. Remember, I said they were bimodal. So the seeds were either gray and round, or they were white and wrinkled. See how they kind of always, there's either one or the other. The flower, like I said, white or violet. Where the flowers were placed, either along the axis or along the end of the stem. Again, bimodal traits in pea plants were a huge reason why they were awesome in terms of the original study of genetics by Mendel. So he started with purebred plants and he crossed the plants with different forms of traits. So again, a purebred means that you have many offsprings that have led to the same exact trait. Again, we are using the word purebred interchangeably with the word homozygous, and we will get to that again in a moment. So in the parent generation, he crosses the tall pea plants with the short pea plants, and he only gets tall pea plants back. So what ends up happening is that the short trait, again, seems to disappear, and he's not sure why. By the way, he did these crosses himself. In order to do this, he did, he was the one fertilizing the plants on this. The F1 generation. So this, right, the tall pea plants that he created become the F1 generation. When the F1 generation cross, Mendel allows them to self-pollinate. The F2 generation that is created is now a mix of tall and short plants, even though none of the parents were short. Again, 3/4 tall, 1/4 short. And this, again, is where we find these large number of offspring very helpful. The higher your sample size is in statistics and probability, the more accurate your findings are. All right, so this is some definitions. A dominant trait, always present in the first generation, and the other trait seems to always disappear. So a dominant trait is the one that's always going to show up in the offspring. If a dominant trait is present, you see it, okay? Recessive traits are the traits that seem to disappear in that first generation cross. They sort of like fade into the background. You don't see them, but we know they still have to be there, okay? So this brings us to the idea of genotype and phenotype. Oh, actually, not quite yet, but sure. The genotype is the actual pair of alleles. These are the actual genes that you have inherited from your parents. Please keep in mind, alleles always come in pairs. Again, we are talking about organisms that are reproducing sexually, which means you get one allele from mom and the other allele from dad, and those two alleles together are what determine your traits. Now, again, those actual alleles that you are inheriting from your parents is your genotype, okay? Actually, let me go back real quick. A phenotype, which apparently I didn't type anywhere on this slide, so for that I apologize. A phenotype is what we actually see displayed. So we'll get back to that in a minute. So a phenotype is what we see expressed on the outside. It is what we can observe, but the genotype is the actual genes present in the organism that they actually inherited from their male and female parents. All right, so this is your little flow chart. Alleles make up genes, genes make up chromosomes. So again, Alleles always come in pairs. You get one set of alleles from mom, you get the other set of alleles from dad, and together they make a pair. So then we have paired up genes and also paired up chromosomes, okay? So this brings us to the idea of dominant and recessive. Oops, did not mean to do that. So again, allele, different forms of a gene. Dominant is the trait that always shows up in the organism if it is present, and recessive is the trait that is masked or covered up if a dominant gene is present. The only time we see a phenotype that is recessive is if there are no dominant genes present. Because remember, dominant is kind of like the loud kid. They sort of take over the space. And it is always going to be visible if it is present. In peas, for example, tall is dominant, short is recessive. All right, so how do we symbolize these? If I have a capital letter, then that is a dominant allele. So if I wanted to show a homozygous tall person, or a tall plant, I should say, then I would type in two capital letter T's. Capital letter is always used for the dominant allele. Again, homozygous or purebred means that you have the same exact allele that came from both parents. Again, lowercase, lowercase, short, short, to recessive alleles. Still homozygous, still purebred, but now we have a homozygous or a purebred recessive plant. So that is where we're going to see the short, because again, the only time I'm going to see short is if it's both homozygous recessive. On the bottom, what we see is a capital T and a lowercase t. So I have one dominant gene, I have one recessive gene, but that is my genotype, capital T, lower key. I got tall from maybe mom, I got short maybe from dad, but I still have that pair. However, my phenotype, what I am going to see in this plant is tall, but it is a mixed set of alleles, so we call this a hybrid set, or we can call it a heterozygous, because I have two different genes for the same trait. But again, the phenotype, what I am going to see is going to be tall because there's a dominant gene present, and whenever there's a dominant gene present, we are going to actually experience or see that expressed. All right, so what are our big ideas here? Big ideas are simple. First of all, alleles and genes are what control your inheritance of traits in organisms. Our phenotype is determined by our genotype. In other words, what we see on the outside expressed is determined by the genes that we actually inherited from mom and dad. Genotypes can be made up of dominant and or recessive alleles for different traits. Again, if it is dominant, that is what we're going to see expressed. The only time we see a recessive gene expressed is if you are homozygous recessive. In other words, if your genotype only has two recessive genes, a recessive from mom and a recessive from dad. In your DNA, the nitrogen bases, remember when we talked about these, when we talked about macromolecules, we had 4 different nitrogen bases. These are what make up our genetic code, and our genetic code is determined by the alleles that we receive from our parents. Again, one from mom, one from dad. These are our traits, and these determine what our DNA actually ends up looking like. One more time, alleles make up genes, genes make our chromosomes, and each human body cell has 46 chromosomes. Not too surprisingly, 23 of those chromosomes came from mom, 23 come from dad, and they end up pairing up. Because again, half of our genes come from the female, other half of the genes come from the male, and when they get together, they make a full set of chromosomes. Now, it's 46 for humans. It could be any number depending on what kind of organism you are, but we tend to focus on humans because we are human. All right, ladies and gentlemen, I hope that helped. And you can now complete your notes. Have a great day.